And what I want to do is I want to start with gram-positive pathogens first. And so basically with this chapter, what this chapter does is it takes a look at some of the most relevant gram-positive and gram-negative pathogens. And a lot of this information, hopefully, we already know, because a lot of it is repeat from chapter 16. And I have in most of the PowerPoints written the page in the textbook from which the information comes, because it's been gathered from several of the chapters in the textbook. So as far as the gram-positive pathogens, the first genus that we're going to look at is the Staphylococcus genus. And you should already know a lot of the characteristics of the Staphylococcus. You should know that it is a gram-positive cocci, that it is arranged in clusters. So gram-positive cocci arranged in clusters. Okay. It is salt tolerant. That is the reason why the mannitol salt agar plate was developed to select for Staphylococcus. The fact that it's salt tolerant also tells us that it can live in dry places. So it's tolerant to desiccation, to dryness. That means that it can survive on objects fairly easy. It is a facultative anaerobe. It is catalase positive. Pyogenic means that it produces pus. There are two main species among the Staphylococcus genus that are often pathogenic, and actually these are opportunistic pathogens: Staphylococcus aureus and Staphylococcus epidermidis. We are going to concentrate on Staphylococcus aureus, which is the more virulent of the two. So Staphylococcus aureus is actually a normal flora in some parts of the body. It is one of the most common causes of nosocomial infections. So going back to chapter 19, if you look at the organisms that most often cause nosocomial infections, among the gram-positives, the number one is Staph aureus. One of the characteristics of Staph aureus that differentiates it from other Staphylococcus is that it can ferment mannitol. Again, that was the basis for the mannitol salt agar plate, which was selective for Staphylococcus, and it could differentiate between Staph aureus, which was mannitol positive, and other Staph that was mannitol negative. One of the features that makes the organism pathogenic is that it has a low infective dose, so only a few cells can cause an infection. So what you do is you need to keep track of the pathogenic factors that these organisms have, the virulent factors. And a lot of these we have mentioned on chapter 16. Remember that Staph aureus had a protein A, which we know from chapter 16 that protein A is an Fc receptor protein, which helps prevent opsonization. So that is a protein A. If the cell can prevent opsonization by antibodies, it can prevent phagocytosis. So prevents opsonization by antibodies. Other features is that often we have Staphylococcal aureus strains that have capsules. They also have leukocidins, and leukocidins are enzymes or proteins that destroy white blood cells, that kill white blood cells, WBCs. Okay. Now, another feature, another enzyme, which is unique to Staphylococcus aureus, is an enzyme called coagulase. So coagulases are unique, only present in Staph aureus. And I'll mention as to what coagulases are here in just a second. Other enzymes that have been identified out of the Staph aureus strains have been hyaluronidases and staphylokinases. As far as toxin, we'll mention a couple of toxins. We'll start with the enterotoxin, which is a toxin that affects the GI tract. This PowerPoint is showing you some of the virulent features of Staph aureus. So it has protein A here, and it shows you how protein A acts protein A is trapping antibodies by the Fc region, preventing opsonization. This is the protein that causes, that is the coagulase, also called a clumping factor. And what coagulase does is it traps fibrinogen, and it is going to help clot blood so that the organism can hide inside clotted blood. This is again protein A, which turns out is a very versatile type of protein. It does bind the Fc region of antibodies to prevent opsonization by antibodies. However, it does a lot more than that. And this kind of gives you a little bit of some of the other features that protein A has. You don't have to know them. It's just an extra information that tells you how pathogenic Staph aureus can be. Often the strains are going to have capsules. It is a polysaccharide capsule, which again is going to protect the organism from phagocytosis in the absence of complement, but also it makes it harder for the organism to colonize in vivo. Leukocidins, we talked about that. These are proteins that destroy white blood cells. Coagulases are important because this is an enzyme unique to Staph aureus. As a matter of fact, it's one of the enzymes that are identified in the organism when we want to identify Staph aureus. Essentially, what the enzyme does is it clots blood. So it helps the organism hide inside clots. So you can take a little bit of plasma, and you inoculate the plasma with the Staph aureus, and in a few hours, the plasma is going to be clotted. Hyaluronidases breaks down hyaluronic acid, which is often present in connective tissue, allowing the organism to invade connective tissue. Staphylokinases are going to do the opposite of coagulases. They're going to break down clots. So the organism has a way of hiding inside clots and then escaping with staphylokinases. Lipases break down lipids. This is another enzyme like coagulase that is extremely important. Beta-lactamases are enzymes that inactivate a whole family of antibiotics called beta-lactam antibiotics. All of the penicillin-based antibiotics are beta-lactam antibiotics. So a Staph aureus strain that has the beta-lactamase enzyme will be resistant to all of the penicillin-based antibiotics, which is one of the largest antibiotic families that we have. Likewise, cephalosporins are going to be sensitive to beta-lactamases. These are also beta-lactam antibiotics. So if the organism has beta-lactamases, several families of antibiotics will be ineffective against the Staph aureus. If that wasn't enough, some Staph aureus have cytolytic enzymes that are going to break down the membrane of cells. Again, we mentioned leukocidins, which are going to destroy white blood cells. As far as toxins, we have mentioned the enterotoxin, which most strains, or at least 50% of the strains, carry. It is a heat-stable toxin that causes food poisoning. And this is your, quote-unquote, common form of food poisoning with nausea, diarrhea, vomiting, and then you pretty much get over it. Now, the really serious toxin, however, is the toxic shock syndrome toxin. Both of these, the toxic shock syndrome toxin and the enterotoxin, are super antigen toxins. So these are a form of exotoxins that stimulate the immune system to become completely unrestrained. A toxic shock syndrome toxin can become systemic and causes toxic shock syndrome. And we'll go back to toxic shock syndrome here in just a second. So we have the enterotoxin, the toxic shock syndrome toxin, and then there's a third toxin called the exfoliative toxin. And as the name implies, this toxin is going to exfoliate skin. It's going to begin to make skin peel away. Staph aureus strains that have the exfoliative toxin cause a condition called Staphylococcus scalded skin syndrome. All right, so you should know the three types of toxins that we talked about in regards to Staph aureus: the enterotoxin, the toxic shock syndrome toxin, and the exfoliated toxin. Now, as far as diseases that Staphylococcus cause, number one, keep in mind that the most common infections caused by Staph aureus are going to be non-invasive diseases and cutaneous diseases. So by far, those are going to be the most common forms of conditions that we get from Staph aureus. As a matter of fact, about 70% of staphylococcal infections are going to be skin infections. Skin infections like impetigo, furuncles, carbuncles, staphylococcal scalded skin syndrome, etc. However, Staph aureus can also cause systemic infections like toxic shock syndrome, bacteremia, endocarditis. So you should be keeping track of the kinds of infections that Staphylococcus aureus can cause, what are the most common infections, and the kinds of toxins that and virulent factors the organism has. So again, most of the Staphylococcal aureus infections are going to be skin infections. Most of the skin infections are going to produce pus. That's a characteristic of the organism. And so we have infections like impetigo, which is an infection of the epidermis of the skin. Cellulitis, which is an infection of connective tissue. Furuncles are infections of hair follicles and sebaceous glands. Carbuncles is many furuncles together, so many hair follicles infected. When it comes to systemic diseases, probably the most serious one, or one of the most serious ones, is toxic shock syndrome, which is caused by the toxic shock syndrome toxin, which can be absorbed into the blood. And the initial signs and symptoms involves fever, high fever, vomiting, sore throat, muscle pain. However, very, very quickly, the patient is going to go into shock, low blood pressure, renal failure, cardiac failure, and death. The disease has been associated with highly absorbent tampons, and because the organism will proliferate in an area that is humid with blood, etc. Other systemic infections: bacteremia, endocarditis, pneumonia. Bacteremia means blood, bacteria in the blood. Endocarditis will be an infection of the lining of the heart, including the valves. Osteomyelitis, especially sometimes in children. We mentioned also the scalded skin, staphylococcal scalded skin infections, and that is due to the exfoliated toxin, which is present in some Staphylococcus aureus strains. These kinds of infections are especially common in young children, so oftentimes we see them in hospital nurseries. And the exfoliated toxin is going to exfoliate the skin, causing it to peel away. Okay, so as far as diagnosis and treatment, the best way to diagnose it is to try to grow the organism from a skin culture, from a wound culture, from a blood culture, depending on the kind of infection the organism is causing. One of the problems that we have with the Staph aureus, and this is a very, very serious problem, is that we have many, many strains, and we have even more strains since every year, that are becoming resistant to antibiotics. Staphylococcus is right now completely resistant to all forms of penicillin. We have to use other types of antibiotics to treat Staph aureus. One of the group of antibiotics that were developed to treat Staph aureus was the methicillin group. Around the 1980s, late 1980s, early 1990s, we began to see methicillin-resistant Staph aureus. We call it MRSA. Right now, methicillin-resistant Staph aureus is actually quite common, unfortunately. So once we get Staph aureus that is resistant to the methicillin family of antibiotics, we have even less of an arsenal to choose from to try to fight Staphylococcal aureus infections. So at this point, what we're left with are more toxic antibiotics, like Sinercid and Cybox, which were developed specifically for resistant Staph aureus. Also vancomycin, which was also developed to eradicate Staph aureus, and the name vancomycin was given because the antibiotic was supposed to vanquish Staph aureus. So these are more toxic antibiotics. We do have nowadays vancomycin intermediate Staph aureus and vancomycin resistant Staph aureus. There are still rare strains, but we're beginning to see this new type of a Staph aureus, which is actually quite scary. So at this point, what we're left with is very, very toxic antibiotics, Sinercid and Cybox, once you have Staph aureus resistant to the more common antibiotics. When it comes to prevention, best prevention is hand washing, especially in a clinical setting, good cleansing of wounds, aseptic use of catheters, appropriate use of antiseptics, etc. So just aseptic techniques. That's all we have to try to prevent staphylococcal infections. Okay, so that essentially gives you an idea of Staph aureus. You should know the most common infections, which are cutaneous. The series of toxins the organism has is foliatic toxin, enterotoxin, toxic shock syndrome toxin, and what these toxins cause, what kind of skin infection or infections, staphylococcal infections these toxins cause. You should also know the virulent factors like protein A, capsules, coagulases, staphylokinases, leukocidins, etc. Okay, any questions about staphylococcus? Okay, and so we're going to move on to the streptococcus. The streptococcus were characterized and classified by a microbiologist called Dr. Rebecca Lansfield. And in the 30s and 40s, she took upon herself the task of identifying and classifying streptococcus. So especially in the medical world, we classify streptococcus based on what we call the Lansfield group of classifications. And so we don't really go by the genus and the species of the streptococcus, we go by the Lansfield groups. And some of the most important Lansfield groups as far as pathogenic streptococcus are going to be groups A and groups B. Some of these streptococcus do not have a Lansfield group, like Streptococcus pneumonia, for example. Now, as a genus, Streptococcus is somewhat similar to Staphylococcus. They're gram-positive cocci. Sometimes they tend to be ovoid. Their arrangement is different. They arrange themselves in pairs or chains. Their oxygen requirement is markedly different. Streptococci are aerotolerant, whereas Staphylococcus are facultative anaerobes. The fact that they are aerotolerant tells us that they are fermenters. They can only ferment sugars, and they are catalase negative. Okay, that's another end result of that's the reason why they're aerotolerant. Okay, so those are the general characteristics. And a good way to distinguish a Staphylococcus from a Streptococcus is the catalase test. That's a really, really important, quick, easy test to identify whether you have one genus or the other. Okay. The Streptococcus that we're going to look at two of them. One of them is the Streptococcus pyogenes, which is actually called in the medical world, it's called group A strep. The other one will be Streptococcus pneumonia. Streptococcus pyogenes is a beta hemolytic Streptococcus. Very difficult to grow. It typically, you grow it in blood agar plate or other forms of enriched media. Most of the pathogenic strains, which appears to be pretty much most of the strains, have capsules. It is normal flora, and we find it in the population at a rate of 5 to 15% of the population carrying the organism. Part of the reason why the number is so low nowadays is because we have been trying to eradicate the organism. It is a pretty bad pathogenic organism with lots of virulent factors. Because of that, a rapid identification test has been developed. It is the rapid strep test. that identifies carbohydrate A found on the surface of group A Streptococcus. So carbohydrate A is a complex polysaccharide. It is antigenic. So during this test, the nurse will swab the patient's throat and then add some reagents that have the antibody against carbohydrate A. And if there is a reaction, that means the patient has a streptococcus. So within a matter of a few minutes, they can know whether they're dealing with a strep throat or not. So this is the rapid strep test. Okay. So the streptococcus is makes very tiny colonies beta hemolytic. That's beta hemolysis. It arranges it into chains, has a bit of an oval shape. The organism is very virulent, but outside the body is actually quite weak. It cannot really cause an infection or compete with other organisms very easily. Part of it is because it doesn't compete well. It's an aerotolerant, so it doesn't. It is a fermenter, so it's going to grow slowly. So it typically causes infection when the normal microbiota are depleted. You need a large inoculum to establish an infection, or when you are dealing with somebody who's immunocompromised. Part of the virulent factors, and it has a lot of them. M protein, which we mentioned in chapter 16. It is a protein that helps the organism break down C3b. And if it breaks down C3b, now it can prevent opsonization by complement. So prevents C3b opsonization. Okay, so with that, it prevents phagocytosis. Another protein is protein G, which is an Fc receptor. That means it is similar to the protein A of Staphylococcus. And again, protein G is going to prevent opsonization, but in this case, opsonization by antibodies. Again, with these, the organism can escape phagocytosis. Okay, this is by antibodies. Okay. Protein F helps the cell attach to the host cell. So F for the fimbria that helps the cell attach. Now this one is a little bit more interesting, a hyaluronic acid capsule. Some strains of group A strep have been found to make a capsule made of hyaluronic acid, which is a compound found in our connective tissue. So by doing this, the immune system looks at the capsule and it doesn't see it as foreign and therefore is not going to attack it. So it's a way for the organism to hide inside a capsule. And again, there's just one strain that has been found with that feature. All right, and that's just showing you the little proteins that stick out of the cell that helps it, gives it a protection from our immune system. The organism also secretes proteases, which break down proteins, hyaluronidases, which break down hyaluronic acid, and that way it can access connective tissue. DNases break down DNA. Streptolysins are going to dissolve clots, and they're also, actually no, not streptolysins, streptokinases dissolve clot. Okay. Streptolysins break down blood cells, especially red blood cells. So this is the reason why the organism is beta hemolytic. The organism also has C5a peptidases, which destroy C5a, which was the complement that attracted white blood cells to the area of infection. By destroying C5a, group A strep essentially prevents C5a from calling macrophages and other white blood cells to the area of infection. Okay. Now, as far as toxins, there's a couple of toxins that this organism can produce. There's a pyrogenic toxin, which is a pyrogenic supertoxin, so it is superantigen, sorry, which means it is damaging to the immune system, creates an exaggerated immune response. The erythrogenic toxin causes a condition called scarlet fever. And some group A strep can produce a streptococcal toxic shock syndrome toxin, very similar to the one produced by Staphylococcus. As far as conditions caused by the organism, the most common condition is going to be a strep throat or pharyngitis. Okay. Some strains may cause scarlet fever, especially the strains that have the erythrogenic toxin. So the organism will need to have the erythrogenic toxin to cause scarlet fever. And not all group A streps have the toxin. Other infections caused by group A strep, otitis media, which is an inner ear infection, middle ear infection, pneumonia, sinusitis. It can cause skin infections like impetigo, cellulitis, pyoderma. It also can cause a streptococcal toxic shock syndrome, just like a staph could cause toxic shock syndrome. Necrotizing fasciitis or myositis is the flesh-eating infection. This is a very, 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 very serious skin infection that could lead to death. Yes. Yes, you do. You do definitely need to remember the diseases that are caused, especially the important ones. Like, for example, strep throat, scarlet fever. This is the only cause of a scarlet fever, is group A strep. Toxic shock syndrome. So we have two organisms that can cause toxic shock syndrome, Streptococcus group A and Staphylococcus. Necrotizing fasciitis is not common, but it is so dramatic that it makes the news when somebody dies of necrotizing fasciitis. This is what we call the flesh-eating bacteria that literally just destroys tissue within a matter of hours. And the only prevention, or the only way to fight it, is to cut limbs and to remove tissue by surgery. So it's a very dramatic infections. And really what it shows you is how pathogenic the organism is. So the answer to yes, you do have to know these infections. There's your impetigo caused by strep throat. So we have two organisms that can cause impetigo, Staph aureus and Streptococcus, a group A strep, which is a Streptococcus pyogenes. This is scarlet fever. This is also scarlet fever. And this is a strep throat, which gives us the pus in the throat region. Necrotizing fasciitis is the really dramatic type of wound infection. So you can read some of the signs and symptoms. It is an invasive condition due to all kinds of enzymes, hyaluronidases, streptokinases, DNases, streptolysins, exotoxin A and exotoxin B, which destroys tissue. Okay. All right. Now, the only good thing, which is one more thing. We know strep throat, kind of, we know that is a pharyngitis, type of pharyngitis, in which we have fever, we have the redness, we're going to have pus in the throat. Pus is important because the organism is a pus former. Some people carry the organism in their noses and in their throats. And again, we're trying to eliminate this organism in these people so that we have less carriers. Transmission is a typical close contact, ingestion of the organism or inhalation. The organism doesn't do well outside the body. So the contact transmission must be really close contact transmission. Okay, now as far as treatment, the only good thing about this organism is that it is not resistant. It is one of the few bacteria that are still very sensitive to penicillin and penicillin-based antibiotics or erythromycin. So treatment is actually quite easy. Just a 10-day course of some form of antibiotics should do it. Prevention, especially with strep throat, any form of pharyngitis that gives fever and pus should immediately be checked out by a physician so that a course of antibiotics can be started. There are no vaccines. And this is the reason why the organism is so scary. Sequelas. Sequelas are conditions that appear after the initial infection. And in the case of group A strep, the sequelas are autoimmune diseases, two of them, rheumatic fever and glomerulonephritis. So these sequelas appear if we have an untreated strep throat. And that's one of the urgencies in treating the disease, as the condition, as quickly as possible and as effectively as possible. Rheumatic fever is an autoimmune condition that damages the heart, cardiac muscle. It is rare in the United States because physicians know about the dangers of group A strep and they try to combat it, and that's so it's not as common. The other sequela is glomerulonephritis. This is an inflammation and an autoimmune condition that destroys the kidneys. So both of these conditions could be deadly. So you should know what sequelas are and what causes them. Okay, questions? Okay, the second streptococcus we're going to look at is Streptococcus pneumonia, which causes pneumonia. It's one of the causes of pneumonia, one of the most common causes of pneumonia. We call it pneumococcal pneumonia. Sometimes it's also called community-associated pneumonia. About 60% of pneumonia cases are going to be caused by this organism, Streptococcus pneumonia. It is a streptococcus, so it is a group A cocci, typically found in pairs, sometimes in chains. It doesn't have a Lancefield classification group. Under the microscope and even on a plate, the organism, the colonies, look very similar to those of group A strep. One of the differences, however, is hemolysis. Streptococcus pneumonia is alpha hemolytic, whereas group A strep was beta hemolytic. So that is one of the important differences between those two organisms when it comes to identification. Strep pneumonia is actually a normal flora in the throat and the mouth. Most of us carry it and it's just fine. We're used to it. It doesn't cause disease on us. There are a few pathogenic strains that may present a problem, but for the most part, we all carry the organism. That's where the source of the alpha hemolysis we see during a throat culture. However, young children may not be protected. Their immune system may not be well enough to protect against the pathogenic strains. Same for the elderly. So pneumococcal pneumonia is especially common in high children and the elderly. Signs and symptoms of the pneumonia: runny nose, is an upper respiratory congestion initially, cough, fever, chest pain, sputum. The chest pain may cause a shallow, rapid breath due to poor oxygenation. The organism is ultimately not as virulent as group A strep. It only has one virulent factor, and that is the polysaccharide capsule that interferes or destroys C3b opsonization. That's all it has. It does have adhesins that allows it to stick to the respiratory epithelium. Diseases that it causes: pneumococcal pneumonia, otitis media. If it becomes systemic, it would cause septicemia, endocarditis, meningitis. That's the other scary thing that it can cause. This is one of the three most common causes of meningitis. Diagnosis: try to grow the organism from a specimen. Treatment, again, is Streptococcus, and for the most part, Streptococcus has remained pretty sensitive to antibiotics. So a course of most antibiotics should take care of the problem. Again, prevention: a large chunk of the population carries the pathogenic form of strep pneumonia. So maybe trying to eradicate that organism, the carriers, would be one strategy for prevention. There is a vaccine available. The vaccine has been made from the capsule of the organism. And even though it is not as effective in children or the elderly, we do give it to children and the elderly. Because it's not a very good vaccine, the vaccine has been conjugated. So if you go back to chapter 18, that means that the vaccine has been joined with an irritant that is going to irritate the immune system and help the vaccine work better. So you need to make a note or a list of organisms for which we have vaccines. And the first one we have encountered is Streptococcus pneumonia. Okay, questions? Okay, moving on, the last of the gram-positives we're going to look at is going to be Mycobacterium tuberculosis. And Mycobacterium tuberculosis is an acid-fast bacteria. It's also called Koch bacillus or the tubercle bacillus. It is the causative agent of tuberculosis. Humans are the only reservoir. And the organism is acid-fast, which means it has a mycolic acid capsule. Cell wall, which is the root of its pathogenicity. So because it's acid-fast, it's going to be a slow grower. Nutrients diffuse very slowly. That also means that it's going to be resistant to chemicals because the chemicals will diffuse into the cell slowly. The organism grows very slowly. It is resistant to drying. Generation time is long. It's about 16 hours when most organisms have a generation time of 20 minutes. Difficult to treat because it grows slowly. Most antibiotics rely on the organisms growing fast. So if the organism is a slow grower, the antibiotics are not going to be as effective. So that's the problem with the organism. Okay, so tuberculosis is a respiratory disease. You know from chapter 19, it can be transmitted by airborne transmission. It is extremely common around the world. It is estimated that 1/3 of the world population has come in contact or carries the mycobacterium. Signs and symptoms. Okay, so the first infection is generally asymptomatic, especially if the patient is healthy. The immune system should control the infection and may or may not eliminate it. If it eliminates it, great, the infection is gone. If it doesn't eliminate it, the infection becomes latent tuberculosis infection. Once a latent tuberculosis infection is established, the infection may or may not move on to cause an active tuberculosis disease. And it may take many, many years before the active tuberculosis sets in. So signs and symptoms is going to be a fever, slow-grade fever, weight loss, night sweats, persistent productive cough meaning that a lot of phlegm comes out, oftentimes with a little bit of blood in the cough. The organism can be inhaled, airborne transmission, or respiratory droplets. The organism is phagocytized by the macrophages in the alveoli of the lungs. However, the cell wall of the organism is going to prevent phagosome-lysosome fusion, and the cell can just remain in the white blood cell, hiding from the rest of the immune system. And these little white blood cells that are infected, right there, are going to be walled off by healthy white blood cells. So this is an early tubercle or a granuloma lesion. So the bacteria sometimes is going to break free from the phagosome, divides inside macrophages. New macrophages are recruited to wall off the infection. And that is what we call the early tubercle or granuloma lesion. Okay. A layer of connective tissue develops around the tubercle, and when that happens, we call that a granuloma or a Gone fossa or Gone complex. If we have lymph nodes involved, we call it a Gone complex. So now we have the granuloma walled off by connective tissue. If the bacteria survives but it doesn't multiply because there is not enough oxygen or the pH conditions are not appropriate, the latent tuberculosis, the infection is established away from the immune system. At this point, the patient is not contagious because the organism is not active. The patient doesn't show any signs and symptoms. The organism is not detected in the sputum. X-rays will be clean because there are no lesions in the lungs. And however, the tuberculin test will be positive. In the tuberculin test, what they do is they inject in the skin a little piece of the bacteria. And since the patient has a latent tuberculosis infection, is going to have antibodies against the bacteria. And when it is injected under the skin, the antibodies will act and create a positive tuberculin test. Yes, that was one of the bonus questions in chapter 18. Chapter 18 and in the last chapter, because if you're exposed to the antigen, you're going to make antibodies. And so what's giving the reaction are the antibodies that are being created upon exposure to the antigen. Yeah, and actually the question was not exactly that. The question was, it said that the person had been vaccinated. So the vaccine introduced the antigen, which meant the person was not immunized. Now it had antibodies. And when the tuberculin test, which introduced the tuberculin bacteria into the person, the antibodies would react against the bacteria introduced. So the tuberculin test detects the presence of antibodies against the bacteria. So it's not telling you have tuberculosis. It's telling you have been exposed to the organism. There has been a primary infection, first infection before the tuberculin test, because the person has the antibodies. The first infection can be a vaccine, or it can be a natural exposure. Okay. So yeah, that's exactly right. So at this point, when we have a positive tuberculin test, the next step is to have an X-ray. And if the X-ray confirms lesions in the lungs, then they may try to find the antigen, try to do a culture to further identify and confirm. So this is latent. So we're talking about latent infection, in which the infection is not active. However, if it becomes active, now obviously we have a bigger problem. An active miliary tuberculosis. Now the organism begins to break free from the neutrophils or the phagocytic cells that have ingested them, and begins to infect other cells. That creates further lesions called caseous necrosis for dead tissue in the lungs. So typically an active tuberculosis patient is going to have a positive tuberculin test. The organism will be seen in the sputum. So when they do an acid-fast stain of the sputum, it will come out positive for the acid-fast bacteria. And if they do an immunofluorescent test, it will be positive for Mycobacterium tuberculosis. So the antigen is there. It's not just the antibody, but the antigen. The patient is contagious. And we can see lesions on the X-ray. So active tuberculosis gives us a contagious patient. So this is showing you the lesions right there and lesions right here, tuberculosis lesions in an X-ray of the lungs. Eventually the lesions can become so serious that actually you have a cavity or completely destroyed tissue in the lungs. The organism could spread to other organs, joints, gallbladder, kidneys, bones, etc., and can cause further infections in those organs. So the miliary tuberculosis is when the disease begins to spread through other parts of the body. Okay. So as far as diagnosis, we have the tuberculin test or Montaut test, which detects for the presence of antibodies indicating prior exposure to the organism and possibly a latent infection, maybe an active infection. So that's when an X-ray is done. And a sputum test could also be done. The X-ray will give us lesions if we have a active infection. The sputum test will detect the presence of the antigen, the actual bacteria, which will further indicate we're dealing with an active infection. Now, when it comes to treatment, this is even more tricky because it turns out the organism has become extremely resistant to lots of antibiotics. Number two, because it grows so slowly, treatment is lengthy. It can be 6 months, it can be a whole year of antibiotics. So you're going to be taking 2 or 3 combinations of antibiotics for an entire 6 months or even 1 year. So it's extremely difficult to stick to the treatment. And that's one of the reasons why we have resistant tuberculosis bacteria, is because people don't stick to the treatment. So you should know it's extremely difficult. Combination therapy given more than one antibiotic for a number of months. Isoniazid, rifampin, ethambutol, pyrazinamide are 4 of the antibiotics given, and it's typically a combination of at least 2 or 3 of these antibiotics. Prevention, okay. Typically, if somebody has been exposed to the organism, we give them prophylactic antibiotics, meaning that we put them on a course of antibiotics, typically rifampin, to try to prevent the onset of the infection. There is a vaccine available, however, we don't use it in this country. It's typically used in countries where tuberculosis infections are more common. So if somebody has been vaccinated with the BCG vaccine, the tuberculosis vaccine, from that point on, the person is going to have a positive tuberculin test. Okay, so you should know those basics about tuberculosis. Questions? Okay. Oh, okay, good. So I'm going to go ahead and proceed to the gram negatives. And what I'm going to do, I want to stop the recording.